This is the body. It's made up of many different complex systems, organs, and tissues. But though these systems may be very different and perform a wide variety of functions, they do all have certain things in common. For example, they are all made up of cells. Cells are the building blocks for everything in our body, and inside each of these cells are a series of organelles that carry out various tasks. Part of a cell's job is to act as a factory to produce one vital molecule in our body, protein. Protein is particularly valuable for things like growth and repair, and without it we would never grow and cuts would never heal. This is why protein synthesis is so important. The video will focus on the first phase of this process called transcription. This process takes place inside the nucleus of a cell. Deoxyribonucleic acid, more commonly known as DNA, is vital for life as it provides all the genetic information that determines an individual's traits and characteristics. DNA looks similar to a twisted ladder. This shape is called a double helix. DNA is made up of molecules called nucleotides. Each nucleotide contains a phosphate group, sugar, and a nitrogen base. There are 4 possible nitrogen bases that a nucleotide may have. These are adenine, represented by A, thymine, represented by T, guanine, represented by G, and cysteine, represented by C. Adenine nucleotides can only join with thymine nucleotides, and cysteine nucleotides can only join to a guanine nucleotide. This can be easily remembered by thinking about apples and trees and cars and garages. The way protein synthesis starts is by a specific enzyme called RNA polymerase binding what is called a promoter section of DNA. This is also often called a promoter codon. Once the promoter has been found, the RNA polymerase will lock onto the DNA strand and begin to break the hydrogen bonds and unzip the DNA. This is similar to unzipping a jumper or a pencil case. The RNA polymerase slowly moves along, separating the strand of DNA, and as it does, complementary free-floating nucleotides come together and use the newly separated DNA as a template to form a single RNA strand. The same rules apply with the nitrogen bases as a normal strand of DNA, but there is one key difference. Instead of the thymine joining with the adenine, adenine joins with uracil. An easy way to remember this is an apple under a tree. This is not only because uracil is energetically less expensive to produce, but also because thymine breaks down once it leaves the protective barrier of the nucleus, where uracil does not. Once the mRNA strand is transcribed from the DNA, it is termed pre-mRNA because it has to be edited further in the nucleus before it can move into the cytoplasm for the next phase of protein synthesis called translation to take place. Once the mRNA strand has been created, all of the introns, also known as non-coding sections, are removed by a process called splicing. This splicing process is performed by a nucleoriboprotein called spliceosome. It works by bringing the 2 ends of an intron section close together to form a loop. The spliceosome then cuts the RNA to release the intron section and join the 2 ends of the exon or coding section of the RNA strand together. Once the intron section has been cut away, the spliceosome then disassembles itself. Once all the non-coding sections of RNA have been removed, all that remains is a section called mature RNA. It is then that the mRNA strand is ready to move out of the nucleus and into the cytoplasm so that the process of translation can take place. So now you know the basics of the complex system transcription. This system is so intricate that unfortunately there was too much detail to cover in this short video. However, if you wish to further understand this amazing process that is a key part of creating one of the most important nutrients in our body, protein, then it is strongly encouraged that you keep researching. If you want to learn more about the next phase of protein synthesis called translation, you can check out the video done by the Protist pals. Thanks for watching!